Hello and welcome. You are listening to a podcast by the Milner Centre for Evolution at the University of Bath. I'm Professor Turi King, your host, and today I'm talking to Andrew Preston, Professor of Microbial Pathogenicity, about his research understanding how bacteria cause disease in humans and animals. So Andy, I'm always really interested in knowing how people came to do the research that they do. So was there something that triggered it for you to get into this area of research?
I wish it was a more conscious decision, but no, I was an undergraduate studying biochemistry. Part of the course was a final year project, so I talked to cancer biologists, all sorts, but ended up in a microbiology lab, and that was that. And I think if you are a researcher, and I say this to some of my tutees and they think I'm mad, I'm not convinced it really matters what it is. It's the process. So if you really are a researcher, you could be a researcher in anything. But in many cases, you don't know anything other than what you've been exposed to. In my case, it was microbiology. And that's what I've stayed in because it rings all the bells for me in terms of what I want to do.
So what is it that you're researching now?
So at the moment, we're primarily looking at a group of organisms called Bordetella, and the most prominent member of that group is pertussis, and it causes whooping cough, which is resurgent in many countries worldwide. So we're asking questions about what is happening, what's the likely trajectory, and of course, can we do anything about reducing the numbers of cases that we're seeing?
So what are you looking at with it?
In terms of broad questions, it's how does Bordetella do what it does? So what's the interaction with the respiratory tract? How does it bind? How does it grow? How does it overcome host defenses? How does the host respond? And I think one of the interesting things which maybe ties in with the Milner is that pertussis is really a very new disease, probably only been around in its current form since the Middle Ages. And even within the last 100 years, we know it's still on an evolutionary trajectory. It's still adapting to the human host. And we've done rather consequential interventions, such as vaccination, which again, even in the last 20 years, has left a strong imprint on this organism. And even since Christmas of this year, we're seeing another change in the way that this bacteria has come back after COVID, and it's doing new things, leading to some pretty large outbreaks and infant deaths in the UK this year.
So how's it coming back after COVID? What's causing that?
Pretty much all respiratory diseases, they depend on people doing what we do now, being close to each other and breathing to transmit. Obviously, that was interrupted significantly during COVID. The actual cases of whooping cough largely disappeared from most countries where you've got good reporting. So we're interested in what happened during that time. We think the world shut down. The world didn't. Rich countries shut down. The rest of the world carried on. And so I think what we're seeing is a reseeding in developed countries of disease from elsewhere, but those bacteria are slightly different from the ones we had pre-COVID. And so we're really interested in trying to track what has happened to see how, if you sort of reintroduce these bacteria into what is largely a naive population, because we haven't had any exposure for a while, being able to sort of compare before and after, because I think we're going to see a different pattern of disease going forward with pertussis. There were signs of it changing prior to COVID. I think this has just really re-imprinted a sort of new host immunity pattern on it, and we're waiting to see how it plays out in the long run.
So how do you do that? How do you study that?
These are population effects, so we can do all sorts of fun and games in the lab looking at the interaction between individual cells, individual bacteria. That's not really the forces that play at the population level. We need these broad pictures of host immunity. So we're trying to work out how to follow these bacteria in people more closely, because at the moment we're utterly limited to somebody reporting to a GP or a hospital with a severe case of whooping cough. That's the notifications we get, and most of our understanding about the way this bug is moving through human populations is by perhaps just relying on those tip of the iceberg notifications. So where's this bug in the human population when it's not causing disease, which we think is probably a relatively small proportion of cases? So who's it in? Is it just within kids? Is it within teenagers? What's happening in the older population? What proportion of the human population might be carrying the bacterium? And that's where probably most of the selection pressure is happening. How long do they carry it? And who's transmitting to whom? So we're trying to develop new studies and new tools so that we're no longer reliant on what is probably a tiny percentage of the cases, which is classic cough.
So this is the thing. So there's vaccines for this. So presumably people will not necessarily go on to develop disease in a big way, but they can still be transmitting it, I'm guessing.
So this is one of the impacts that the human intervention has had over the last 50 years. So the first set of vaccines were whole cell vaccines. You literally grow up a massive vat of bacteria, you kill it and inject it. And the advantage for that is it displays a whole range of targets for your immune systems, thousands of targets, because that's the nature of a whole bacterium. During the late 1990s, we switched to a subunit vaccine, so 5 highly pure proteins from the bacterium for reasons of safety and reactogenicity of the vaccine. That's created an entirely different focus for the immune system. So whilst those vaccines we know have done a great job of preventing very young babies from becoming seriously ill, those vaccines actually don't stop people from picking up the bacteria in their nose. They don't cause disease. So in all of the high-income countries that have switched to those vaccines, we suspect the level of circulating bacteria has grown and grown and grown and are giving rise to these very significant periodic outbreaks. So vaccination is critical for saving the lives of young babies. And unfortunately, we've seen 8 deaths in the UK since Christmas of this year, showing it's still a potentially fatal disease. But we're unsure as to the ability of these new vaccines to continue to control the circulation of the bacteria. And that's the big question in the field at the moment, is how do we implement control? Because although we could probably, in fact many have designed new improved vaccines, it's very difficult to implement a new vaccine to replace one that already exists, particularly for the childhood schedule, where it's crammed and most vaccines are given as, say, 5 or 6 in one shots. So the science is there, the regulatory framework, and how to pick apart and replace, one part of a complex schedule is the big question at the moment.
So if it's sitting within people, presumably it's evolving as it's sitting there. So what's the selection pressures that's going on?
So it's interesting. So, we have this picture of organisms, and bacteria in particular, because they can evolve quickly, constantly changing and picking up mutations. We don't see any of that with pertussis. So if you compare the range of mutations, the number of mutations, it's what's described as monomorphic. So we can sequence, and we have done, in fact, hundreds of strains, and we see just a handful of mutations between them. So we're not sure whether that means mutations don't arise very quickly, so maybe it has a really high level of, editing out these mutations, or whether the selection is so strong that any mutations that do arise create a fitness effect so that they're quickly outcompeted. But the one thing we do see is that in those countries that have switched into new vaccines, one of those vaccine antigens, one of the proteins used is an outer membrane protein, sits on the surface of the bacteria. And we know from studies that most of the strong immune response is directed against that protein. And so in most countries that have switched to those new vaccines, 90% of the strains no longer express that protein. So that's clear evidence that there is selection on these bacteria. But it doesn't fit that all of the selection on these is from vaccines, because actually we vaccinate mainly babies, and we know that the immune response to those vaccines wanes, so it's literally gone by the age of about 10. So part of those studies that we need to do is to work out where is the bacterium. The epidemiology doesn't fit with it residing and circulating solely in young babies. So who is carrying it, for how long, and what is that interaction? Because it's an organism, it's a bacterium that is remarkably devoid of the diversity that we would expect to see in something that's just, as far as we know, circulating solely within the nose of people. Biology 101 suggests you have to vary in order to constantly circulate amongst the same host, because you would expect them to build up some level of immunity. So we think the organism has an ability to suppress long-lasting immune responses, and we would like to work out how it does that.
So is it something that mainly affects, so we know young babies, is it something that it tends to affect older people and that kind of, adults generally tend to be okay with it? Or is it something that we all need to be starting to be concerned about?
You've hit the nail on the head. So if you look at a clinical microbiology textbook, whooping cough is a toxin-mediated disease of young babies. But actually, if you look at studies where they've tried to look for evidence of what might be causing chronic cough, and thousands of people reported to GPs and hospitals around the world with undiagnosed chronic cough, so 3 weeks or more is the classic one. In some of those surveys, up to 30% of those people have evidence of recent pertussis infection. So the emerging picture that in very young babies are the ones that display the classic symptoms of whooping cough, so this paroxysmal cough with the large intake of breath because they've been coughing, i.e. the whoop. But actually that's probably a feature of a very poorly developed small respiratory tract, i.e. babies. In older people, adults, adolescents, it's probably manifest just as a prolonged chronic cough. And certainly in the outbreak we're seeing at the moment, where there's over 1,000 cases a month, over 50% of those are in 15-year-olds or older. And most of those are showing as a really annoying, debilitating but chronic cough rather than classic whoop.
So it's almost like it's hiding, isn't it? Because you know a whooping cough and you're kind of looking for a certain sort of set of symptoms. But actually what we're finding is it's manifesting itself in a different way in older people. And so it's clearly travelling and on the rise and causing infection.
It is. I think one of the unknowns there is whether we've been duped by the initial description that pertussis infection was whooping cough, nothing else, and it happened in babies. And if you don't look for something, you don't find it. So actually, I think the jury is out on whether it's always infected older people, but without the classic whoop, or whether this is a newer manifestation. And it's probably actually will be a mixture of both. So I think the more the organism is circulating because of the change in vaccines, I think probably the more you will see it causing symptomatic infections in older people. And interestingly enough, it's changed within just decades. It's undergoing a metamorphosis, and probably 3 or 4 different manifestations since we've started to really A, track it, and then B, try and do something about it by vaccinating.
So is that essentially what you're trying to do? You're trying to track this and sequence it and see if there's any kind of particular differences that are arising? How are you doing your research?
So we do use genomics as sequencing because it's so devoid of clear signals. So we can't just focus on a subset of genes and know that we can pick up and we'll be able to classify lineages or clades or things that you can do maybe for, say, E. coli or Klebsiella. We've realised that to be able to give fine-scale resolution and to work out maybe transmission chains, or really to identify differences that distinguish strains that were isolated pre-vaccine or during the different types of vaccine use, we need whole genomes. Even then, the signal that we get is a low level of mutations, and we're still trying to work out what those mutations actually mean. Do they change the biology in somehow? I think that loss of pertactin, so that outer membrane vaccine antigen, there is very clear evidence now that gives the organism the greater ability to evade vaccine-induced immunity. But there are other certain types of mutations, single changes in bases, that have gained almost mythical status because the only signal we've had of, for example, the change during the whole cell vaccine era, but actually you get different results depending on the assays you try to look. So are they more toxic, for example? Some studies say yes, others say no. We're limited by tools to really devise assays that are relevant to the symptoms we see in people, because there is no assay for cough, for example. So whereas maybe with Staph aureus, they cause different types of infections, go into the skin or into the blood, with pertussis, all of the clinical data is you get cough, and that's it.
So what does evolution tell us about pertussis then?
To me, it's a really interesting case for being able to almost do the sort of time travel experiments. So the genomics has told us actually that pertussis has evolved really very recently, maybe even within the last couple of thousand years, which evolutionary-wise is virtually moments ago, from an ancestor that's still around today. It's called Bronchoseptica. So Bronchoseptica is a bacterium that infects the respiratory tract of pretty much any mammal in which you care to look, you will find it. But it's very rarely a pathogen, so it's largely just something that it sits in the nose. It can be carried for the lifetime of an animal, but then transmits to others, but is perhaps more of a commensal than a pathogen. Pertussis is almost a subset of Bronchoseptica that's undergone an evolutionary process where actually it's streamlined its genome. So it's got rid of maybe 25% of the genome of its ancestor, and in doing so has become exquisitely adapted to only infecting the nasopharynx, the nose of humans. And it's outcompeted, so it's eliminated its ancestor from that niche. We no longer really find Bronchoseptica in people. And so we're really interested in that process as an exemplar of evolution to really become highly adapted. And of course, what it's done during that adaptation is it's changed its biology. It's now become a pathogen. Maybe it needs to be pathogenic, i.e. cause disease and symptoms to spread more efficiently. And of course, the fascinating side of that, it's probably done so in combination with changes in its host. So many of the acute respiratory pathogens of humans, such as influenza, are really quite new. So for most animals, they exist in relatively small populations. And so an organism has to maybe be carried for a long period of time by that host, keeping that host relatively healthy and therefore mobile, for it to be able to then encounter a new host for transmission to occur. If you now pack your host into dense populations with a high replacement, i.e. birth rate, you can afford to just let rip and be highly pathogenic, outcompete other organisms in your niche. And of course, you're going to have a constant supply of new hosts. So you can kind of not really care what you do to your host once you've moved on. And so that's the perfect breeding ground for these highly transmissible acute diseases that is a hallmark of humans. Influenza, and I think pertussis is one of those. If we can sort of compare Bronchoseptica, and interestingly, there's another subset, parapertussis, that's somewhere in between pertussis and Bronchoseptica. It's even more recent. It causes whooping cough, but less often. We're trying to do studies, look at the three. What do they bind to in the nose of the different hosts? How has pertussis become so much better adapted to the human nasopharynx? What are the specific interactions that enables it to outcompete? And then the response that it induces in people, does that give us clues about this evolution of disease from what was largely a sort of commensal existence? And so we think we've got a great model system to do some really sort of nice time travel experiments, hopefully find out something useful along the way about could we actually interrupt that process, because obviously if we can stop pertussis from binding to the nose, it can't establish infection and therefore can't cause disease.
So that's really interesting because, as you were saying, so it sounds like it's super adapted. So you're looking at adaptation in bacteria, and in pertussis it's become super adapted to be able to infect us through our noses. So what other bits of bacterial adaptation are you looking at the moment?
You could almost say pertussis has got this new trait, this ability to cause disease. And I think when we think about bacteria gaining new traits, we think that they've acquired genes to do so. This is a fascinating example. It's lost genes. So we know that the difference between a pertussis genome and Bronchoseptica is it's just jettisoned 25% of its genome, maybe getting rid of things it doesn't need anymore, because we think Bronchoseptica possibly can also survive in the environment outside of a host. But it's jumbled its genome to do so. So it's got largely the same genes as Bronchoseptica, but got them in different orders in the genome. And we're really interested in how does that play a role in the ability of a bacteria to generate adaptation, and whether that's relevant to what's happened. If we focus just in on what's happening in the respiratory tract, we can do those experiments. We can get respiratory tissues, we can grow respiratory cells, and just actually look at that interaction between the three different species and the cells from the different hosts. And that's what we're doing at the moment. We're trying to identify patterns of which molecules from the bacteria bind to what's on the host, how do they differ between them, and then how does the host cells respond differently to each of the three species, and does that then set up the consequences that we see, i.e. disease in one or benign carriage from another.
So what's next for you?
I think the next big thing will, can we get together at a global level and convince funders that what we need to do is establish exactly where the bacterium is in the human population. So a large-scale study of carriage. So can we identify the cohorts that are carrying it? So, what proportion of people might carry this bacterium at any one time? Is it particular age groups? How long do they carry it for? What percentage might even have symptoms? How often do they transmit? Because I think what we do need to really do is to work out how to use our vaccines effectively. So this idea of boosters. So in the UK, we now offer a booster shot against pertussis to pregnant women. because we know that the really young babies who are too young to start their own vaccinations are the ones super susceptible to the fatal disease. But actually, we need to know whether we need to cocoon older people. So grandparents have a lot of interactions with young babies. So do we need to add more boosters? Will that help curtail the resurgence that we're seeing? But how often can we boost people and it still be effective? And who's going to be the most beneficial groups to receive those boosters? And I think that's a pressing situation that has immediate public health consequence, but is a remarkably difficult one to implement globally where it needs to be done, because we see different patterns of disease in different countries. And who's going to pay for it is always the big question.
Andy, thank you so much for talking with me. This was a podcast by the Milner Centre for Evolution at the University of Bath. I'm Turi King, and thank you for listening. If you have any thoughts or comments on this or any other episodes, please contact us via our X channel at Milner Centre. For more information about the Milner Centre for Evolution, you can visit our website.